Hello everyone and welcome to Master Microbiology today. Today I'm talking to you about some bacteriological terms, some terms that are commonly used in bacteriology. Number one of these terms is bacteria. What is bacteria? Bacteria is a microscopic unicellular organism that is prokaryotic as opposed to eukaryotic. Number two is bacteriology. What is bacteriology? Bacteriology is the study of bacteria and their relationship to medicine, agriculture, and industry. What then do you mean by bacteriophage? Bacteriophage is a virus that can infect bacteria. So, a virus that can infect bacteria specifically is referred to as bacteriophage. There are other names that this bacteriophage is called by. When we say phage virus, that means bacteriophage. Another term here is bacillus. Bacillus is a rod-shaped bacterium. Bacteremia. Bacteremia is the presence of bacteria in the blood. When there is bacteria in the blood, that is when we refer to as bacteremia. What about bacteriuria? Bacteriuria has to do with urine, and it means the presence of bacteria in urine. Bacterial. Bacterial. What do you mean by bacterial? Bacterial means when something is caused or related to bacteria. So anything that is related to bacteria or caused by bacteria is called bacterial. Then we have bactericidal. Bactericidal. It is a substance that can kill bacteria. Bacteriostatic. Bacteriostatic. Static means just to stop something, to halt it. And bacteriostatic is a substance that can inhibit the growth or the multiplication of bacteria. Then we have cocci. Cocci is a spherical bacterium that can exist as a single cell. It can be in pairs, it can be in chain, or it can be in clusters. If it is occurring singly as a cocci, it is called micrococci. If it is occurring in chains, it is called streptococci. If it is occurring in clusters as in grape, it is called staphylococci. Now, let's talk about Vibrio. Vibrio is a curved rod-shaped bacterium. A curved rod-shaped bacterium is referred to as Vibrio. Then Spirilla. Spirilla is a spiral-shaped bacterium. So, a spiral-shaped bacterium is referred to as Spirilla. Now, let's define spirochetes. Spirochete is a spiral bacterium with a very tight coil. Actinomycetes is the next one. Actinomycetes is a gram-positive filamentous bacteria with a branched growth pattern. Let's continue. When we mention abiotic, abiotic is a non-living. Abiotic means non-living. Abiogenesis. Abiogenesis means generation of life from abiotic components. That means from non-living components. Actinomycetes or actinobacteria is group of filamentous bacteria. Adhesion. Adhesion means attachment of microorganisms over biotic or abiotic surface. Agar. Agar is a polymer of galactose sugar, that is agarose, and agaropectin, which is derived from algae, and it is used to solidify culture media. Agar medium. Agar medium is solid or semi-solid culture medium containing agar. Agar diffusion. It is diffusion of molecules through matrix in agar medium, and it is used during antibiotic sensitivity test or during some other similar test. Agarose gel. Agarose gel is a semi-solid matrix formed by the solidification of agarose. It is mainly used in the differentiation of proteins and nucleic acids. Aerobes. These are microorganisms requiring atmospheric oxygen for respiration, for survival. Anaerobes. Anaerobes are microorganisms that can survive in presence. Beg your pardon. Anaerobes are microorganisms that can survive in the absence of atmospheric oxygen. Aerotolerant. Aerotolerant are anaerobic bacteria or anaerobic microorganisms that can tolerate atmospheric oxygen. Antibiotics. Antibiotics are medicines or chemicals used against bacteria or bacterial infections. Antibiotic resistance. Antibiotic resistance is the ability of bacteria to escape the effect of antibiotics. Antifungal. These are medicines or chemicals which are used against fungi or fungal infections. Antifungal resistance is similar to antibacterial resistance or antibiotic resistance. It is the ability of fungi to escape the effect of antifungals. So, when fungi escape the effect of antifungal drug, it is referred to as antifungal resistance. Antimicrobials. These are medicines or chemicals used against microorganisms or microbial infections. Antimicrobial resistance, AMR. It is the ability of microorganisms to escape the effect of antimicrobials. Anthelmintic. Anthelmintic. These are medicines or chemicals used against helmintic infections. Antiprotozoal. These are medicines or chemicals used against protozoa or protozoan infections. Antiparasitic. These are medicines or chemicals used against parasites or parasitic infections. Now, let's look at the next one, which is antiviral. Antiviral are medicines or chemicals used against viruses or viral infections. Antiviral resistance is the ability of viruses to escape the effect of antivirals. Then we move to the next one, which is antiseptics. Antiseptics are antimicrobials applied over living tissues. Archaea or archaea. These are domain of prokaryotic organisms, similar morphology to bacteria, but unique biochemistry against microorganisms. Aseptic, sterilized or free from microbes. Aseptic is an area which has been sterilized or an area that is free from microbes. Asepted. Asepted. These are microbial cells lacking septa. Lacking septa. Agglutination. This is clumping together of different particles. It can be blood cells or other things. Agglutination, clumping together. Antigen. An antigen is a foreign body capable of activating the immune system. Antibody. Antibodies are glycoproteins produced by the immune system against a specific antigen. Antagonistic, showing inhibitory sidal response to another. Amphitrichus. These are bacteria with a single flagellum at each end. Atrichus. Atrichus, lacking flagella. Alpha. Hemolytic. Microorganisms partially disrupting red blood cells, giving greenish zone of hemolysis. Attenuate. Attenuate means to reduce the virulence of a pathogen. Autoclave. Autoclave is a sterilizing equipment that uses a pressured hot steam to sterilize using an autoclave. Usually, an autoclave sterilizes materials that are put in it at 105 kilopascal and it will run for 15 to 20 minutes. Autotroph. Autotroph is an organism that synthesizes their own food. ATP, adenosine triphosphate, energy currency of cells, stored form of energy in a living cell, in the living system. Antimicrobial, antimicrobial sensitivity testing or antimicrobial susceptibility testing. This is a procedure to test the sensitivity of antimicrobials. ATCC. This is the American Type Culture Collection. It is an organization storing and providing standard microorganisms. Antibacterial resistance. When we talk about antibacterial resistance, we are referring to ABR. ABR is antibacterial resistance, while AMR is antimicrobial resistance. Now, I want to move on to microbiology terms that are starting with the letter B. The first one on the list is bacterial culture. This is bacteria grown in a culture medium. Bacteriology is a study of bacteria. Bacteriophage is a virus that can infect bacteria. Bacillus, those are rod-shaped bacterium. Bacteremia, presence of bacteria in the blood. Bacteriuria, presence of bacteria in urine. Bacterial cause or related to bacteria. Bactericidal, substance having the capability to kill bacteria. Bacteriostatic, substance having the capacity to inhibit bacterial growth or multiplication. Basal medium, culture medium with minimum nutrition that is enriched with highly nutritious substance to make enriched special media. Biohazard, biological components that are hazardous. Broth, BROTH, liquid culture medium. Broad spectrum antibiotics. Antibiotics acting against a broad range, both gram-positive and gram-negative bacteria. Biochemical tests. A set of tests to identify the biochemical characteristics of a microorganism. Biochemical test is a set of tests used to identify microorganisms based on their biochemical characters. Biofilm. A biofilm is a thick film aggregate of bacteria formed within a thick, slimy extracellular matrix. Bioluminescent bacteria. These are bacteria emitting light. Blood agar. Enriched culture medium containing blood. Blood-borne infection. These are infections transmitted through blood. Buffer. Solution that resists change in pH of a solution. BSC, Biosafety Cabinet. I move to other terms that are starting with the letter C in microbiology. The first one on my list is capsid. Capsid. Capsid is a proteinous capsular shell enclosing viral genomes. Capsule. Capsule is gelatinous extracellular layer covering bacteria, usually made of polysaccharide. Capsulated, having capsule as the outermost layer. Carbon source, nutrient component in a culture medium that provides carbon for microbial growth. Chain, arrangement of microbial cells in form of a line. The next one is chitin or chitin, protein in the cell wall of the fungus. Chromosome, bacterial genome DNA. Cidal effect means killing of bacteria or microorganisms. Cilia. Cilia is fine hair-like appendages in eukaryotes. Ciliates. Ciliates refer to protozoans that have cilia. Clone. Offspring of a single microbial cell is a clone. Cluster. This is the arrangement of microbial cells in a group. Cryopreservation. This is the preservation of microbial cells in a very low temperature. Coagulase. Microbial enzyme that can clot plasma. Coagulase test. This is a test to detect the ability of a microbe to produce coagulase. Coliform. A group of gram-negative bacilli found in the feces of vertebrates. Colony. Visible group of microbial cells developed after microbial culture. Usually the bacterial colony or any other colony of microorganisms are found on the Petri plates using the appropriate media. Complex media. Media that supports the growth of broad range of microorganisms is referred to as a complex media. And as an example, in bacteriology, neutral media or LB nutrient media or LB agar or nutrient agar usually support the growth of all forms of bacteria. So we now move on to talk about compound microscope. This is a light microscope that uses two sets of lenses, the objective lens and the eyepiece lens. Conjugation. Conjugation is the transfer of bacterial DNA by direct contact between two bacterial cells. Contamination. Contamination is the introduction of unwanted microorganisms. Contaminated growth. Contaminated growth. Microbial culture with unwanted, uninoculated microorganisms. We can also call it contaminated culture. Now, talking about co-culture or consortia. This is mutual growth of two different species, genera of microbes in a single culture or in a single media. Commensals. Commensals are also referred to as normal flora. Culture counter. Culture counter is a device to count microbial cells in a suspension. Culture. Culture means to grow or to multiply microorganisms. Culture medium with grown microorganisms. Culture again. These are isolated and multiplied single species of microorganisms. Culture medium. Nutritious substance that supports the growth of microorganisms. So, nutritious substance. So substances that can serve as food for the microorganisms are referred to as culture medium. Now we'll move on to the next one, which is CFU. CFU is referred to as colony forming unit or colony formation unit. These are microbial cells that form a single colony. So that is where I'm going to stop with microbial terms that start with the letter C. We will move on to microbial terms that start with letter D now. Welcome back. The first letter that starts with letter D is decontamination. Decontamination means making a sample or object sterile. That is the removal of microorganisms that are alive. Another one is decomposers. These are microorganisms that degrade complex organic matters into a simpler form. Desiccation. Desiccation means the removal of water. Diatoms. These are microscopic unicellular algae. Dinoflagellates. Dinoflagellates are unicellular algae with two flagella. Diffusion. This is spreading over a matrix of medium or gel. Usually the spreading is from an area of higher concentration to an area of lower concentration. Diplococcus. This is arrangement of microbial cells in a group of two. Diphtheroid. Diphtheroid is a commensal bacteria morphologically similar to C. diphtheria, similar to Corynebacterium diphtheriae. Dilution. This is a suspension of microorganisms or chemicals in a certain proportion. So you suspend it in a certain proportion. So if you have a solution or a microbial suspension of a particular concentration, you can dilute it by adding sterile distilled water such that the concentration you're going to have at the initial state is different from the concentration you have when you added water or when you diluted it. Direct microscopy. This is counting of microbial cells in a sample using a microscope. Disinfectant. These are chemicals that kill or inhibit microorganisms and is used in inanimate surfaces. An example of disinfectant is ethanol. Usually 70% ethanol is used to sterilize the workbench after and before microbiological work is done on a bench. Disc, 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 is a small circular, usually 6 mm filter paper piece mostly used to carry antibiotics for tests or chemicals for tests. DNA, that is deoxyribonucleic acid. Enterobacteriaceae or enterobacteria, bacteria found in gastrointestinal tract, members of the Enterobacteriaceae. Electrophoresis. This is a technique to separate macromolecules and nucleic acids using electricity over agarose gel. Endotoxin. Pyrogenic lipopolysaccharide, mostly within cell wall of gram-negative bacteria. Toxin within the bacterial cell. Endo, inside. Then when we say exo, outside. Enterotoxin. This is an exotoxin affecting intestinal cells. When we say entero, it means intestine. Enterobacteria, intestine. So enterotoxin is a toxin, exotoxin affecting intestinal cells. Now, what do we mean by exotoxin? These are toxins released outside the microbial cell. Remember, endotoxin is released inside, while exotoxin is released outside the bacterial cell or the microbial cell. Enteric fever. This is typhoidal or paratyphoid fever. And when we say enteric, it affects usually the area of the intestine, is affecting intestinal cells. Now, let's look at the definition of eubacteria. Eubacteria is a domain of prokaryotes, including true bacteria. Extremophiles. Extremophiles are organisms thriving in extreme environments. ELISA, capital letters. This is enzyme-linked immunosorbent assay. It is a detection technique. It's an antigen-antibody reaction technique. So you can use ELISA kit for different tests, especially in virology. ETC is electron transport chain. ESBL, extended spectrum lactamase, a type of lactamase enzyme. Now let's look at microbiology terms that start with letter F. The first one is facultative anaerobe. This is anaerobe that can even survive in the presence of oxygen. Normally these are anaerobes, but they can survive in presence of oxygen. Facultative aerobe, aerobic microorganisms that can even survive in anaerobic condition. Now, let's look at the definition of fermentation. Fermentation is an anaerobic breakdown of organic substances such as sugars into alcohol, acid, or gas by microorganisms. If you want to demonstrate fermentation based on this definition, you get about a few grams of sugar, maybe 500 grams, in a beaker, or not in a beaker, in a small jar that has a lid. In a small jar that has a tight cover, you pour about 1 liter of water in it, or 2 liters of water in that jar, then add your 500 grams of sugar. After doing that, add yeast, just about 5 grams of yeast or up to 10 grams of yeast, and then you shake it and leave it to stand for 3 to 5 days. After 3 days, by the time you check back, the sugar in the fermenter, which is your jar with the lid, will have converted to alcohol by the action of the anaerobic process, converted by the action of the yeast. So it will be converted into alcohol, some into acid. As it stays longer, the conversion also goes on. So by the time you open the lid, you see that gas is the first thing you're going to meet. Now let's look at the definition of flagella. Flagella are hair-like protruding appendages in the microorganism that provide motility. Flagellates. These are microbes having flagella. Filamentous. Long thread-like morphology of microorganisms. Fimbriae. Fine proteinous appendages, smaller than flagella. Fluorescence microscope. Microscope that uses a fluorescent dyed sample and sample emitted light to form a magnified image of the sample. Fungus. Kingdom including prokaryotic organisms with chitin as a component of the cell wall. Let's move on to G, letter G. Gene. Functional unit of DNA encoding for a particular trait. Genetic analysis. Study of genetic material is referred to as genetic analysis. Gene mapping. Determining the position of genes in DNA is referred to as gene mapping. When you determine the position of a gene in DNA, what you're talking about is gene mapping. genetically engineered or modified, organisms whose genes are artificially modified. Genera or genus is a taxonomic category below a family that constitutes phylogenetically or morphologically similar species. Genotype: complete genetic constituent of a microorganism. Germinal stage: this is a stage of microbes in which they can replicate, they can reproduce. Germs: microorganisms that can cause disease, infection, or that are pathogens. Germ theory: a principle that defines the relation between germs and disease. Gliding bacteria: bacteria that have gliding motility. Globulus: these are antibodies. Gram stain: gram stain is a compound stain composed of crystal violet, iodine, and safranin, used to differentiate bacteria based on the type of cell wall they have. So the bacterial cell wall can be gram positive and it can also be gram negative. That is the gram stain. Gram staining: a staining technique that uses gram stains. Gram positive: bacteria that stain positive or violet after gram staining. Gram negative: bacteria that stain red or pink after gram staining. Growth medium is also referred to as a culture medium. Come back. Now we're looking at the definition of heterotrophs. Heterotrophs, or we can also call them heterotrophic. These are organisms that depend on others for food or for nutrition. This is opposed to autotrophic. Autotrophic do not depend on others. They can make their own food by themselves. So green plants usually are autotrophic, while non-green plants or non-green bacteria or non-green microorganisms are heterotrophs. Halophiles: these are salt-loving microorganisms. Microorganisms that love to stay in an environment that has salt are referred to as halophiles. Then we can also have what is called halotolerant. These are microorganisms that normally don't like salt. They are not halophiles, but if salt is present, they can tolerate it. They can live where salt is. That's why they are called halotolerant. Hazard: hazard means a great risk, and there are many microbiological hazards. Some of the microorganisms that you work with in the microbiology lab are hazardous. They are risky to health, so that is why you have to be careful how you handle them. Hemolysis: hemolysis means the breakdown of red blood cells. Hemolytic bacteria is bacteria capable of breaking down red blood cells. Host: a host is a living body that provides nutrition to parasites. Hyphae: hyphae is thin, elongated, and nucleated branching fungal cells, either septate or aseptate. Now, let's move on to what is meant by immunogen. Immunogen is an antigen or infectious agent. When we talk about immune, that means somebody is resistant or somebody cannot be affected by a particular organism or a particular disease. Immunogenic. Immunogenic means something that is capable of stimulating the immune system. That is referred to as immunogenic. Then, when we talk about immunity, that is resistance against infections or against disease. Immunization is also referred to as vaccination, and it is a process whereby a particular trait or kind of that particular microbe that normally we call disease. So the microbe is injected into a man in a state by which it cannot cause the real disease, so that that individual can have a taste of it. So that taste the individual has of it, the next time the main disease is coming, the body's defense will shoot at it more than when it was introduced in its mild form. Now, let's talk about immunodeficiency. This is the lack of ability of the immune system to resist infections. Immune compromise. Immune compromised is a suppressed immune system, just as the name goes, that the immune system is compromised, so it cannot uh, do its work of immunity. Immunoglobulin are antibodies. In vitro. In vitro means in the laboratory. It may also means outside the natural habitat. It also means in the test tube. In vivo. In vivo means in natural habitat. Indicator organism or indicator. These are microorganisms that indicate the presence of certain pathogens without actually isolating the pathogen. I used to tell students that when we talk about indicator species, you know that the principal or the director or the head of department is around. Without seeing him, how do you know? You see where he parks his car, and his car is rightly parked there. So the car is an indicator. The car is not the HOD, but the car is an indicator that the HOD is around. And in microbiology, we have what is referred to as the concept of indicator species. Specifically, we use Escherichia coli as indicator of fecal pollution. So the vibrio or other organisms like Salmonella, we will not bother to isolate them. But the moment we see Escherichia coli in water, we know that that water has been contaminated with feces. So we discard the water or we change our water source. Infection. Infection is invasion of tissue by one or more pathogenic organisms and establishment of disease. Infectious, capable of causing infection. Inhibition, interference and suppression of microbial growth or multiplication, that is inhibition. Inhibitory zone, a zone of inhibition formed around an antimicrobial substance. Invasive, invasive means it's capable of penetrating tissue layer. Inoculation, inoculation is introduction or transfer of microorganisms. Inoculate, to introduce or to transfer microorganism into something that did not have that particular microorganism previously. Inoculating loop. Inoculating loop is a tool with a loop at its end that is used to transfer microorganisms, especially bacteria. For fungi, transferring of fungi, we use the inoculating needle. So, because fungi, you have to cut the hyphae. Inoculating loop is not strong enough to cut the hyphae, so we use inoculating needle to cause fungal hyphae, while we use inoculating loop to pick bacterial colony from one point to another point and for other uses. Inoculating wire, tool with a straight wire that is used to transfer microorganisms. So this is inoculating wire, as I said, inoculating needle. Inoculum. Inoculum is a substance or sample with microorganisms to be tested. Incubate. This is to keep or maintain in ambient conditions for microbial growth. So when you incubate, you are keeping it at a particular temperature so that it can grow. Different microorganisms have different temperatures for growth. So the incubator is temperature regulated. You can put the temperature to 37, 40, 25, or thereabout, depending on the growth condition based on temperature. Usually, most bacteria grow at 37 degrees C, while most fungi grow at 25 plus or minus 1 or 2 degrees Celsius. Now, let's look at what is meant by incubation. Incubation means keeping inoculated media in ambient temperature and atmospheric conditions for microbial growth. Microbiology terms that start with the letter J, junk DNA. This is when we talk about junk DNA. Junk DNA is DNA whose function is not known. Now, let's move on to what is meant by Koch's postulates. Koch postulates is the criteria to define a microorganism as causative of a disease. Now, let's move on to the next one, which is lab, LAB, capital LAB. Lab simply means lactic acid bacteria or bacteria that produce lactic acid. Laminar airflow cabinet or hood. This is device used to prevent contamination of and from sample. Lancefield classification. This is classification scheme of Streptococcus species based on serological characteristics, surface antigens, etc. Latent, existing without showing symptoms. That is what we mean by latent. It is not showing symptoms, but it may have been infection may have started, or the infectious agent may be in a place but is not yet showing or eliciting any symptom. That is referred to as latent or latent infection. Latent infection without observable symptoms. Then lethal, lethal, potential of taking life. Lethal dose, LD, amount of toxin that is enough to kill. Lichen, obligate association of fungus and algae. Light microscope, microscope that uses visible light is referred to as light microscope. Liquid media, blood culture medium in a liquid state. Lysogeny, lysogeny is a type of life cycle of viruses or bacteriophage using viral genome, integrated bacterial DNA for multiplication or replication. LPS is lipopolysaccharide. At letter M, microbiological terms that begin with letter M and on and on we go. Talking about McFarland standard solution. This is a solution of 1% barium chloride and 1% sulfuric acid at a fixed concentration. Methanogens. Methanogens are microorganisms producing methane gas during metabolism. Metabolism. These are chemical reactions occurring inside living cells. Mesophilic. These are microorganisms whose ambient growth temperature is between 20 to 45 degrees Celsius. They are also referred to as mesophiles. And most microorganisms occur at this temperature. And this temperature is the temperature range of the human body also. So this is why most microorganisms also do are mesophiles, they affect man. Microbiology itself is the study of microorganisms. Microbes also means microorganisms. Microbia relating to microorganisms. Microorganisms, organisms that are visible only under a microscope. Microscopy is the use of microscope to produce a magnified image of microscopic objects. Microscope device used to form a highly magnified image of tiny objects for clear visualization. Microbial flora, microorganisms normally living in on our body surface without causing disease. And there are many organisms that are microbial flora or also called normal flora of different parts of the body. Like our mouth has normally staphylococcus. Our skin normally has staphylococcus. And some other parts of the body normally have some organisms that reside in them. And they do not cause disease. Microaerophilic, microorganisms that can survive in presence of a very low concentration of oxygen. Mold, filamentous fungi or fungi with mycelial body. Monoclonal, originating from a single cell. Mutation, Sudden change in gene is referred to as mutation, and the mutation can be point source and it can also be non-point source. Mutant, organism with certain mutation making it different from their parents. Mycelium, elongated branching structure of hyphae. Mycology, study of fungus. MDR, capital multi-drug resistance. MIC, minimum inhibitory concentration or minimum inhibitory condition. This is the minimum concentration of antimicrobial agent required to inhibit microbial growth. MBC is minimum bactericidal concentration, minimum concentration of antibiotics necessary to kill bacteria. MPN, capitals is the most probable number, is a technique used to estimate viable microbial concentration in a sample. Now, let's go on to look at nosocomial. What do you mean by nosocomial? Nosocomial means hospital-borne or acquired. Now, what do you mean by non-culturable bacteria? Bacteria that can't be cultured in vitro in culture. In fact, microbiologists have indicated that more than 90% of bacteria are non-culturable. Non-selective media. Culture media that support the growth of a wide range of media and produce similar colonies for different microbial species. Negative bacteria. These are gram-negative bacteria. No growth, lack of microbial multiplication, and formation of visible colonies. Now let's go on to look at microbiology terms that start with the letter O. Obligate parasite. These are parasites that can't live outside the host. They need the host. It is a must that the host will be present for this obligate parasite to survive. Then we have obligate anaerobe. These are organisms that can't survive in the presence of oxygen. They can only survive when oxygen is absent. They can only survive in anaerobic environment. Obligate aerobe, on the other hand, are organisms that can't survive without oxygen. That means oxygen must be present for obligate anaerobe to survive. Oncogene. Oncogene. This is a gene capable of triggering mutation in the cell, causing development of cancer. Oxidizing bacteria. These are bacteria that oxidize certain components or substrates. Now, let's look at the definition of a parasite. A parasite is an organism that depends on the living body for survival. So we have what is referred to as parasite-host relationship. So the parasite is living on the host and is harming the host, while the host is not getting any benefit from the parasite. Parasitic, depending on others for survival. So as we talk about parasites, parasite depends on the host for survival. So parasitic means depending on others for survival. Pasteurization. Pasteurization is a technique of sterilization by rapidly heating and cooling the sample or liquid. And this method was developed by the scientist referred to as Louis Pasteur. Pathogen. A pathogen is an organism that has the potential to cause infection. Pathogenic. Pathogenic is capable of causing disease or infection still. Pathogenicity is capable of capacity of the pathogen to cause disease. It's referred to as pathogenicity. Pathogenesis. Order of development of a disease. How the disease progressed, how it started, how it continued, and how it is still continuing. So the history of that disease from start to finish is what we call pathogenesis. Pathology. This is the study of disease. Pathoval. Bacterial strain with similar characteristics except in their pathogenicity. Petri dish or Petri plate is transparent cylindrical plate for containing culture medium in which microorganisms are cultured. Phage, bacteriophage. Phenotype, observable morphological characteristics. Phenotypic, based on observable characteristics or characters. Photosynthetic microorganisms, microorganisms capable of conducting photosynthesis, or microorganisms that can photosynthesize. Usually they are green in color, or they have the green pigment. Pillar, this is short, thin, flexible, and soft bacterial appendages. Pipette, instrument that is used to measure and transfer small volume of liquid sample. Plasmid, they are circular, double-stranded extrachromosomal DNA of bacteria. Now, this definition says that plasmids are circular, but there are a few plasmids that are not circular. So let's take note of that. Majority of plasmids are circular, but not all of them are circular. Presumptive test. This is biochemical test to presume the presence of specific microorganisms in a sample. Probiotics. These are variable microorganisms that are consumed to promote gut flora and gut health. Prions. These are small proteinaceous infective agents lacking nucleic acid. Pseudohyphae, arrangement of yeast cells giving the appearance of a hyphae. Psychrophilic, microorganisms thriving in cold. So microorganisms that can live in cold environments are referred to as psychotrophic or psychrophilic. PCR is polymerase chain reaction. It's a molecular technique to amplify nucleic acid. Now, let's move on to Q, microbiological terms that start with the letter Q. The first one on my list is quorum sensing. This is coordinated behavior or communication in microbial bacterial community. Quantitative analysis or quantitative test. Test to identify and count the number of microorganisms in a sample. Qualitative analysis or quantitative test. Test to identify microorganisms in a sample. Qualitative does not involve counting. Now, let's move on to the next one, which is rapid test. Test used to quickly identify an unknown microorganism is referred to as rapid test. So these are tests used for quick identification of an unknown microorganism. Recombinant DNA, a chemically combined DNA containing DNA portions of different organisms. Replication, production of exact copies. Respiration, breakdown of an organic molecule inside the cell producing ATP. Restriction enzyme, bacterial enzymes that cleave DNA at specific sites. Reverse transcription, process of the formation of DNA from RNA. RNA is ribonucleic acid. RIA is radioimmunoassay. Now, let's talk about what we mean by a sample. Sample means specimen or materials from where microorganisms are supposed to be isolated. So the sample can be water, it can be food, the sample can be blood, it can be urine, it can also be wound, it can be any surface, it can even be air. Selective media. These are media that selectively allow the growth of certain species of microorganisms while inhibiting the growth of the others. Septate. Septate means that they are divided, and then we have this septate hyphae and aseptate hyphae in fungi mycelium. Serovar. These are serotype bacteria or viral strains with all the characters same but different antigens and stimulate a host immune system for the production of different antibodies. Sheeted bacteria. Bacteria arranged in filaments covered with a mucilaginous sheet. Slime layer. Slime layer is an outer slimy layer in some bacteria made of polysaccharides, glycolipids, and glycoproteins. Species. Species is a taxonomic category below the genus that can interbreed. Spirochetes. Spirally twisting shaped bacterial growth. Sporulation. Process of formation of spore. Spore. A spore is a thick-walled dormant stage of bacteria or fungi that can develop into a viable individual. Now, what do you mean by sterilization? Sterilization is the process of removing or killing all viable microorganisms. Strain. Variants of the same species resulted due to certain variations, mutations in their genes. Subtype, that is strain. Subspecies, a taxonomic category below species including organisms that are morphologically distinct but can interbreed. Staph is Staphylococcus. Strep is Streptococcus. SEM, scanning electron microscope. Now, let's talk about Taq polymerase. These are bacterial polymerase enzymes derived from Thamus aquaticus, and this Taq polymerase must be kept in cold because they can be inactivated. Thermophiles. These organisms that thrive in temperatures above 45 degrees Celsius. Thermotolerant. Organisms whose ambient temperature is below 45 degrees Celsius but can even survive and grow in temperatures above 45 degrees Celsius. So when we talk about something tolerant, it's like normally my food is rice and beans, but in the absence of rice and beans, I can also feed on plantain or on banana. So you get it. My main food is rice and beans, but if rice and beans is not present, I can use others. I can tolerate it. Transcription. This is the process of formation of RNA from DNA. Translation. Process of formation, decoding proteins from RNA. Transformation. This is transfer of genetic material directly from one bacterium to another through a cell membrane. Transduction. Introduction of foreign gene DNA into bacterial DNA via virus. Transgenic. Organisms whose genetics have been altered by incorporation of foreign gene or DNA segment. Transmission. Transfer of pathogens from one host to another. Transposition. DNA, that's transfer of DNA gene from one side to another. That's what we mean by transposition. Transposition. Transmission, transposition. Then TEM, transmission electron microscope. Now, let's talk about urease test. Urease test is a biochemical test to detect the production of urease enzyme by bacteria. Move on to the next one, which is variants. Variant is a subtype, usually of viruses and bacteria, that are genetically different, but the genetic difference is not sufficient to classify as a separate strain. Viruses. Viruses are self-replicating, obligate parasitic infectious agents having only one type of nucleic acid as their genome. Virulence. Microorganisms' ability to damage hosts or cell components and to cause disease. Virulence factors. These are molecules secreted by or expressed within pathogens that enable them to establish infection. Vaccine. This is medicine that stimulates the body's immune response to protect and prevent infection. Variable living. Virology, study of viruses. Winograsky column. This is an instrument acting as a self-supporting ecosystem used to study microbial diversity of an environment. Xanthophyta. This is yellow-green algae. Yeast. Single-celled fungi. ZN stain, also called Zell-Nielsen stain. Staining procedure used to stain acid-fast bacteria, that is mycobacteria, and the acid-fast bacteria cause tuberculosis. So this stain is used to detect if somebody has tuberculosis in a way. Then we have zoonosis, infections transmitted by animals. Zone of inhibition. This is a clear zone without visible microbial growth around antimicrobial components. So this is where I'm going to draw the line at this time in this video. If you have any question or comments for me, my name is Dr. Olubengade Titun, and I'm your host on this YouTube channel. I am a master of microbiology, a doctor of microbiology. If you have any question relating to any of these terms or any other terminology that you want me to help you to talk about, you can contact me by dropping your questions or comments in the comment section below. I want you to like this video, share the link of this video with your family and friends, and then drop your comment and questions in the comment section below. Wow, you have not subscribed? What are you waiting for? Go ahead now and subscribe to this YouTube channel. I am very certain that you are going to be glad you did. Just one dose per day, one per day of all these microbiology terms, and then what else? You are going to become a guru. From a guru to a guru. I'm going to talk to you in the next one. Until then, this video that is popping up by the side for my channel is the next you should go out and watch for now. I'll see you in the next one. Bye bye.